this protein as chromoprotein. Chromo means color. Chromoprotein. Okay? So these are some examples of conjugated proteins. Another class of protein is derived protein. You won't get it in the nature as degraded product of protein. We are eating proteins every day because we need protein for various functions, lots of functions. Immense importance of protein. Among all nutrients, protein is having the highest value in regard to its function. That's why we take protein every day in various forms. As simple protein, as conjugated protein, as derived protein, various forms. These are complex. Proteins are complex, colloidal, big macromolecular compound. They cannot be absorbed as such. So they need to be converted into simpler form, simplest form rather, that is into amino acids. Now these amino acids are readily absorbable from the intestine into the blood. So our system should have a process mechanism by which the proteins can be, different forms of proteins can be made into simpler form. That's why we are having a digestive system. Let us see how these proteins are digested, how these complex things are digested, simplified, and absorbed. Sometimes we absorb the native protein and it creates problem like allergy. Body produces antibodies against these proteins. These allergens, these are proteins. They are not digested. When these proteins are absorbed, they produce antibodies in our body. So it's next time when you will be eating or it will be entering through food, it will create allergic reaction. Somebody cannot eat prawn. They cannot eat lobster, prawn. Because for someone, they may create allergy. Similarly, brinjal. Some cannot take brinjal, cannot eat brinjal. So these are the reasons. Young infants, newborn baby, this baby was given colostrum. Why? We know the colostrum is very good, essential for newborn baby. Why? It will provide immunity. It will provide immunity. But this colostrum is a protein and it is not digested because the inverse digestive system, it is unable to produce, it cannot digest protein properly, all the proteins. That's why only it can digest milk. milk protein. No other protein it can digest. But during this period, for a short period of time, protein can be absorbed, particularly the colostrum system. After few hours, then again it will be unable to absorb protein. The gamma, what is there in in colostrum? Gamma globulin. So gamma globulin is a protein. So this is as such absorbed in our body. So these are the two occasions. One is allergen and one is colostrum. Otherwise, in all other cases, protein must be digested into simplest form. So every day we are taking protein through our diet, and these proteins are of two sources. This protein comes from two sources. One is animal source, another is plant source. The proteins of animal source, A-class protein, having high biological value. First-class protein. Their digestibility, good. No problem in digesting them. We can get all the essential amino acids from them. And in good amount. These essential amino acids we cannot prepare. So we have to take it from outside sources. And the vegetable plant proteins, these are also good. But not like animal protein. So they are actually second-class protein, or you can say incomplete protein. Since all the amino acids, though may be present, but not in adequate amount, which can satisfy our requirement of all the essential amino acids. So that's why every vegetarian has to depend on animal protein. Which animal protein? Milk. I am not suggesting them to eat egg, to eat meat, fresh poultry items. But milk can provide all the essentials. Best is the unique one. Milk proteins also like egg, also very high, having high biological value. So our diet, of vegetarian diet, should have at least 400 to 500 ml of protein, I mean milk. The proteins usually we take through our diet. What are these? Animal protein. What are these? Meat or fish or poultry item. Milk and vegetable. Rich source of protein is beans. It may be soybean, it may be any other beans. Any beans are having good rich source of protein. So your diet should have good amount of protein. You should eat good amount of dal, lentils, any other dal, any other beans. So our diet may contain all these proteins. So now question is how they are digested. First of all, this protein should be denatured first. Then it will be easily digestible. Denaturing means the protein in its structures are having different bonds. They are having different structures, primary structures, secondary, tertiary, quaternary structures. These quaternary structures are broken through cooking. See what happens to the egg white. When we boil it, it is denatured. It is denatured, then again it is easily becomes digestible by the enzymes. So you see it is first, that's why we cook food. By cooking, we are denaturing the proteins. Okay. So one nice preparation is made by our cook. Sometimes we are acting as cook, or our whole-time cook is our mother. So they prepare nice dish, so we eat, relish it, and now let us see how these are digested. So when you will be masticating the food, relishing the food in your mouth, okay, so this food will be mixed with salivary juice. Can you tell me whether salivary juice contains any proteolytic enzymes? Yes. What are these? Lipase? Morega ghusa. What happened to you? Not studying? Hello? Are there any digestive proteolytic enzymes? No. What? Proteolytic?
Enzymes are present in the salivary.
Whether present in salivary juice. Can you name it? Calicri. The amino acids we get in the intestine after digestion, these amino acids actually comes from two sources. Who is talking? Come, I'll give you the mic. Come forward. Two sources. One is exogenous, through food, dietary. Another source of this amino acid is endogenous, from our body itself. Almost equal to the amount of dietary protein. These amino acids come from endogenous sources. Now I want to ask you a question. We are liberating so many enzymes, and you know these enzymes are protein actually. What happens to this enzyme? Whether they are recirculated, coming back to the organ, coming back to the gland for again secretion into the GI tract, or what happens to them? Lot of proteins, lot of enzymes are secreted through different juices. Taurine, it's also a protein. Pepsin, protein. Renin, protein. And all the enzymes of pancreatic juice, these are proteins. All the enzymes of GI tract, thacus and entericus, proteins. So they are liberated into the lumen. So what happens to these enzymes after their action? Whether they are recirculated, absorbed, as such, and again secreted? No. They are also digested. They digest, they are also digested. And when these proteins are digested, they will also produce some amino acids. So these are endogenous amino acids. Moreover, we learned in our GI tract, the mucus lining, mucosal epithelium, these epithelial cells are not permanent. They are not permanent. They will die. They will be displaced from there, from the belly. Now what will happen to them? They will be digested like flesh. So from them, we will be getting amino acids. So these are the indigenous sources. So daily, we have to take protein, or again, one of the purpose, one of the function, to produce those enzymes. Unless you eat the protein, from where these secretory cells will produce these enzymes. They should have raw sources, amino acids. So with these amino acids, they will be preparing these enzymes. Okay? To digest all these proteins, whatever the source, endogenous or exogenous, we need proteases. Proteases, protein digesting enzymes. These proteases are of two types. Endopeptidase, and another type is exopeptidase. Endopeptidase and exopeptidase. What is the function of endopeptidase and exopeptidase? What are the functions? Endopeptidase, simple. One peptide linkage, peptides, amino acid, all these amino acids are linked together by peptide bonds. So among these peptide bonds, these peptide bonds are within, and that peptide bond and that peptide bond are at terminal. These are, and the last lady, she is having one peptide linkage. These are terminal peptide bonds. So our peptide proteases are of two kinds, as I said. One is endopeptidase. They will break the inner peptide bonds. That too, suppose he is one of the amino acid, so he is having amino group, carboxylic group. With this amino group, he is making one peptide bond. With this carboxylic group, another peptide bond. So the carboxylic group is producing one, so that will be broken by the endopeptidase. And example of this endopeptidase, pepsin, trypsin, chymotrypsin, elastase, gelatinase, endopeptidase. So these are enteropeptidases. So these are endopeptidases. And there are exopeptidases. Some of them from pancreas it is secreted. Carboxypeptidase. Procarboxypeptidase. The juice contains procarboxypeptidase. So it hydrolyzes the terminal peptide bond. Suppose she is having one free carboxylic group. Her right hand is carboxylic group. With the left hand, she is making one peptide bond. So this is the free carboxyl group. She is the amino acid having free carboxylic group and she is making one peptide bond. That peptide bond will be hydrolyzed by carboxypeptidases. Okay? So, again we will get from the intestinal mucosa, they are having aminopeptidase. So, aminopeptidase, so mechanism of action of carboxypeptidase and aminopeptidases. Okay. So, now we are prepared with, our system is prepared with two kind of proteases, aminopeptidase, terminal peptidases, that means amino or carboxylic peptidase, and these are exopeptidases and endopeptidases. So whatever protein is coming except this protein, these are not digestible. Okay. Other proteins, digestible proteins, edible proteins, so they will be digested by these two type of weapons. Okay. So when food is masticated, we are relishing or it is mixed with salivary juice, so proteins will not be digested here. Since significant proteolytic enzymes are absent in salivary juice. Protein digestion will be starting from stomach. Because stomach glands, the chief cells, the peptic cells, they secrete pepsinogen. They also can secrete another type of proteolytic enzyme, but these are absent in adults, these are present in infants, renin. The infants drink milk. That milk has to be digested. So, for digesting this milk, they are having renin. With this renin, they precipitate the milk protein, casein, into calcium paracasinate. Okay? Insoluble sodium paracasinate. Now this will be digested by the pepsin. So before pepsin can act, that should be this protein is actually secreted as proenzyme, pepsinogen. This is not activated. So this should be first activated. How these are activated? By the hydrochloric acid secreted from parietal cells. So they will split near about 40 amino acids from one terminal, and after removal of these, you are unwanted, so get lost and be hydrolyzed into amino acids. The other part, remaining part, will be acting as pepsin. Now it is an endopeptidase. It will now split the inner peptide bonds and it will produce short peptides, short of having a few amino acids. This sort of peptides it will produce. Okay? And this is produced, the pepsin can only act in acidic medium and its isoelectric pH is 2. So at this pace, it shows maximum activity. Okay, so after digestion of protein, not completed, only short peptide bonds are produced, peptides are produced. Now they will be further, and some proteins come in. Hare. So pepsin will start acting, and what it will produce? The native protein, it will be digested by it will split, this enzyme will split the inner bonds. As a result, what will be the product? Peptones and smaller peptides. Peptone and smaller peptides. And there will be all the proteins may not be converted. Some proteins may remain intact. Sometimes the plant proteins particularly, they escape digestion. So in stool, we may find some protein, but these are not animal protein. These are actually plant protein. 99% of the animal protein are digested, and 95 to 98% of plant proteins are digested. Remaining part is not digested. Beside this undigested protein, we may get some other proteins. These are what? These are dead cells of the inner lining of mucosa, that is of colon, rectum. There is no proteolytic enzymes. So what will be happening? These cells will be coming through the stool, and these cells will have some protein. When we will be estimating the protein quantity of stool, so we will also estimate those cells, dead cells, those dead cells. So what are the sources of proteins in the stool? Undigested plant protein and the dead cells. The dead cells from intestinal mucosa, dead cells from esophagus, dead cells from stomach, they will be digested because they will make contact with the proteolytic enzymes. They will face the proteolytic enzymes. But in the colon, there is no proteolytic enzymes. So they will escape digestion. Understood? Okay. So protein digestion completed in stomach. So thereafter, the chyme containing all other undigested or partly digested protein will enter into the intestine. Now this chyme will face enzymes of pancreas. We know in the pancreas there are different proteolytic enzymes. These are what? Trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase, progelatinase. So these are the proenzymes. Lot of proenzymes are secreted from pancreatic juice. So all these juice, when it will be entering in the duodenum through ampulla of Vater, these enzymes, proenzymes, will face one enzyme that is secreted from the intestinal wall, and that is called what? Other day you answered it. How trypsin is activated? Entero? So this will first activate trypsinogen into trypsin. Now trypsin is activated, and it is the responsibility of the trypsin to to convert all other pro-enzymes into their active form. Now, pro-carboxypeptidase will be converted into, by the action of trypsin, will be converted into carboxypeptidase. Pro-elastase will be converted into elastase. Chymotrypsinogen will be converted into chymotrypsin. Trypsinogen will be converted into trypsin. Initially, trypsin has to be activated first by enterokinase. And after activation of trypsin, this will activate all other proenzymes into their active form. Now everybody is ready to digest the proteins, their respective proteins. Elastin we are eating through meat, through flesh, we are eating elastin. So elastin will be digested by the elastase. So they will form peptone and peptides. Gelatinase. We are eating gelatin, so that we have shown one day how gelatin and elastin forms this layer. Okay, so they are also digested. Gelatinase also digested into peptone and smaller peptides. Similarly, chymotrypsin and trypsin, they are endopeptidases. All these are endopeptidases. They will split the proteins into peptones and smaller peptides. Okay. Now, there is some difference between the action of chymotrypsin and trypsin. Not similar. Endopeptidase, these amino acids are not alike. They are different. So, all these amino acids are having carboxylic group and forming a peptide bonds. So, the trypsin, it acts on particular type of peptide bonds or particular type of amino acids. This is not identical for chymotrypsin. Chymotrypsin has their specific target, say aromatic amino acids. So aromatic amino acids, they are have carboxylic group. So those carboxylic group are forming peptide bonds, they will be breaking those peptide bonds. So in this way, their mode of action is different. Otherwise, what's the use of two endopeptides? One peptide is enough. So these endopeptides differ in their function, mode of action. It is my recipe, I am trypsin, so I'll choose, oh, this is my target amino acid, so I'll break this. That one is my target amino acid, I'll break this. That one is my target amino, I'll break this. But for chymotrypsin, its target is different. So they will break, say, basic amino acids. The basic amino acid will form peptide breakers. So their target is that. So ultimately, what we will get? We will get smaller peptides like tripeptides, dipeptides, amino acids. Carboxypeptide is when they will be splitting this one, so amino acid will form. Suppose these are the tripeptides. So carboxypeptide will eliminate this one, so amino acid plus dipeptide. Now intestinal mucosa has dipeptidase. Now this dipeptide will break this one, so ultimately 3 amino acids we got by the action of carboxypeptidase, amino peptide or dipeptidase, tripeptidase. So in this way, all the proteins are digested into amino acids. So after complete digestion of protein, what is the product ultimately? Amino acids. Now let us see how these amino acids are absorbed. These amino acids are of two types, L form and D form. L form and D form. D form, those who are having amino acids who are having D form, they are easily absorbed. They don't require any carrier. Just simple diffusion, they can enter into from intestinal lumen, they have to enter enterocytes in the mucosal lining. So first into the enterocyte. So they can enter into the enterocyte by simple diffusion because concentration of D amino acid is higher here, but within the cell is less. So by simple diffusion, they will enter there. But what about L amino acids? Lipoform amino acid, what is the fate? They are actively transported. They are actively transported. And it is transported by co-transporter. Sodium dependent. Sodium concentration is high in the lumen. So because of higher concentration of sodium, it will enter into the enterocyte. Higher concentration to lower. Where it is low? Within the enterocyte. Where it is high? In the lumen. So from lumen, sodium will try to enter because of concentration gradient. The glucose will say, "Bye, please take me with you. I cannot enter without you. So okay, come." So it will accompany glucose along with this. So it is a co-transporter. So these amino acids, all these amino acids will be transported into the enterocyte from the lumen by a co-transporter. Now there are different kinds of co-transporters. There are co-transporter for neutral amino acids. What are neutral amino acids? And it is having one amino group and one carboxylic group. Carboxylic group is the acidic group and amino group is the basic group. Number of basic group when in a particular amino acid, number of amino group and number of carboxylic group are equal, then it is neutral. Understood? And acidic, number of carboxylic group is more than one, but number of amino group is less in relation to carboxylic. This will be acidic amino acid. Similarly, what will be the basic amino acid? More amino group, less carboxylic. So this is simple as that. So for them, there are different co-transporter. For neutral amino acid co-transporter. For basic amino acid, co-transporter. For acidic. For neutral amino acid, there are two types. Again, two types of co-transporter. For neutral, large, small. Large, small. So large neutral amino acid co-transporter, small co-transporter. So with this co-transporter, hey, tell me the time. See these, see aminopeptides. Peptide chain. This is having free amino group here. So this is the peptide bond will be broken, hydrolyzed by aminopeptidases. In this way, different dipeptidase acts on it. So there will be two amino acids. Tripeptidase. So this is the free carboxylic group here. So it will break this. Then this will be liberated. This will be digested by dipeptidase. So in this way, peptide bonds are hydrolyzed. See, this is important. Pepsin, hydrolysis of bonds formed by carboxylic groups. PHM is phenylalanine. So this amino acid will be having one carboxylic group. So this carboxyl will form one peptide bond. Pepsin will break the carboxylic peptide bond of phenylalanine. Wherever it will find phenylalanine, and so here is the phenylalanine. Which one is your carboxylic group? This one, right hand. So break it. Similarly, it will break tyrosine. So you look for tyrosine. Tyrosine, which one is your carboxylic peptide bond? So this one. Okay, break it. So again, TRP means tryptophan. Another kind of amino acid. And MET, methionine. Okay, so this way pepsin can break a long peptide bonds into small peptides. Trypsin, it can act, see the difference. Trypsin can act on arginine and lysine because arginine lysine content is high. So it will break all these carboxylic bonds of these amino acids. And chymotrypsin, see the difference. See the difference. Trypsin and action of trypsin. Write it down. Write it down. How does the action of trypsin differ from chymotrypsin? Chymotrypsin breaks the carboxylic peptide linkages of phenylalanine, tyrosine, tryptophan, valine and leucine, leucine. Whereas trypsin can hydrolyze the carboxylic bond of arginine and lysine, but the content of arginine and lysine is maximum in a peptide chain. So their action is much more. See, this way, elastase, alanine, glycine, serine. Okay? Carboxy peptidase, two types, A and B. B carboxy peptidase, terminal basic amino acid. And she should be basic amino acid. Then she will be broken by the carboxy peptidase B, and A will break the terminal aromatic amino acids. D amino acids, we said they are simply diffuse, but their rate of diffusion is very poor, slow. So absorption of D amino acids is poor. But absorption of L amino acid is the active one, but its absorption is fast. D amino acid, simple diffusion, L amino acid, more rapidly and absorb, active process. See the mechanism of action. These are amino acids. These are amino acids. This is sodium. Sodium amino acid co-transporter. So when these are diffused into this through this co-transporter, sodium is diffused into the because of concentration gradient. Concentration of sodium is in the intestinal lumen. This is the intestinal lumen. Here sodium concentration is high. So because of this sodium concentration high, it will diffuse into this with the support of this transport protein, sodium co-transporter. So this co-transporter will also transport amino acid into this enterocyte. Okay, so this is the co-transfer. These amino acids, it may be basic, it may be acidic, it may be neutral. So depending on that, this transporter and protein will vary. Okay, ultimately what will be there? Sodium will enter here, and more sodium ion and more amino acid will be accumulated here. Now this amino acid will be passing through this by facilitated diffusion. Facilitated diffusion by the process from here, this will be diffused into the blood. So from blood it will be carried away. And what will happen gradually, if sodium ion is accumulated within the cell because of this, what will happen? The cell, what will happen? Gradually there will be accumulation of sodium. So water will also be accumulated, so it will be inflated. So it will lose function. So this sodium has to be sent back into the ECF, extracellular fluid, by the sodium potassium pump. Now the sodium will be pumped. The sodium entered from here to here will be pumped out of the cell into the ECF. From ECF it will be entering into the blood. Okay. Similarly, from blood, potassium ion will be entering into the ECF. From ECF it will be entering within the cell to maintain the balance. So okay, so this is sodium potassium pump. Canometer holo, why this has to be pumped out? Because if it is not pumped out, there will be accumulation of sodium within the cell, so it may lose its activity. So by entering from here to here, it is facilitating absorption of amino acids. Once it is absorbed, now it is easily facilitated by this protein into absorption will be into the blood. Okay, so this extra sodium ion will be pumped out. This way amino acid is absorbed. So absorption of amino acid is simple. There is another path by which amino acids are transported. This is called Mr. cycle. This is called, again it is energy consuming process. See, see, this is the membrane, and there is a membrane protein, gamma glutamyl transpeptidase. So with this, what happens? These amino acids are sent with the help of this protein. It is sending amino acid into the enterocyte, and on entering the enterocyte, it combines with glutathione, and by the action of this enzyme, this glutathione and this amino acid combines. Okay, then it forms glutamyl amino acid. Now this amino acid is separated from this compound. So this amino acid entered into here with the help of this enzyme. But this process requires 3 ATPs, one here, and one here. Okay, so this is gamma glutamyl cycle. With this cycle, you need not mention all this process. You can just mention one molecule of amino acid can be transferred with the help of this enzyme through Mr. cycle or gamma glutamyl cycle into the cell. But this is energy consuming. For absorption of one amino acid, it requires 3 ATPs. So it is a very expensive process. So okay, that's all.